Hello, everyone. I hope you are doing well. In this video, you will learn about the culture medias using in microbiology, classification of culture media, components and raw material of culture media. I hope you will learn a lots of things from this video. You get good information from this video, then please like, subscribe and share this video. Culture media. Culture media are the base where microorganisms can grow with the help of nutrients and under the influence of physical growth parameters. All microorganisms cannot grow in a single culture medium, and in fact many cannot grow in any known culture medium. Some organisms cannot grow in artificial culture medium are known as obligate parasites. Examples: Mycobacterium leprae, Rickettsias, Chlamydias, Treponema pallidum. Based on the consistency nature of the media, they are classified into solid, semi-solid and liquid. Solid media contains agar, 1.5 to 2.0%, as solidifying agent and suitable for isolation of bacteria and determination of colony characteristics. Examples: nutrient agar, BAP etc. Semi-solid media also contains agar, but the percentage content is less, 0.5%. This media is useful for determination of motility of bacteria and cultivation of microaerophilic bacteria. Liquid media that do not contains agar except for required amount of nutrients. This media is useful for propagation of large number of organisms. Fermentation studies and various tests. Sugar fermentation test. Synthetic, defined, culture medium. Composition, contains precise amounts of pure chemical compounds. Known components. Every component and its concentration are known. Consistency, provides consistent results due to its well-defined composition. Application, used in research to study the nutritional requirements of microorganisms. Examples: minimal media like M9 or Davis and Mingioles. Synthetic medium, non-synthetic, complex, culture medium. Composition, contains complex ingredients like extracts of plants, animals, or yeast. Unknown components. Exact chemical composition is not fully known. Nutrient-rich, provides a wide range of nutrients, supporting the growth of a variety of microorganisms. Application, commonly used in clinical laboratories and for routine cultivation. Examples: nutrient broth, tryptic soy broth, and blood agar. There are various types of culture media based on the applications as well as media compositions. General purpose media, basic media. Basal media are simple media that supports most non-fastidious bacteria and are used for the isolation of primary microorganisms. Peptone water, nutrient broth, and nutrient agar are considered as basal medium. Enriched medium, addition of extra nutrients in the basal medium in form of blood, serum, egg yolk etc. to make the medium enriched. They are used to grow nutritionally exacting bacteria. Examples: blood agar, chocolate agar etc. Selective and enrichment media. They are designed to inhibit unwanted contaminating bacteria and help to recover pathogen from a mixture of bacteria. Selective media are agar-based and enrichment media are liquid. Inconsistency, addition of antibiotics, dyes, chemicals, alteration of pH etc. and makes a medium selective. They are of various types like 1. Thayer-Martin agar. It is used to recover N. gonorrhoeae. Contains antibiotics, vancomycin, colistin and nystatin. 2. mannitol salt agar and salt milk agar. They are used to recover S. aureus, contains 10%. Sodium chloride. 3. Potassium tellurite. This medium is used to recover C. diphtheriae, contains 0.04%. Potassium tellurite. 4. McConkey's agar. It is used for Enterobacteriaceae members, contains bile salt that inhibits commonly gram-positive bacteria. 5. Pseudosol agar or cetramide agar. It is used to recover P. aeruginosa, contains cetramide, antiseptic agent. 6. Crystal violet blood agar. It is used to recover S. pyogenes, contains 0.0002% crystal violet. 7. Lowenstein-Jensen. This medium is used to recover M. tuberculosis, is made selective by incorporating malachite green. 8. Wilson and Blair's agar. It is used for recovering S. typhi, is rendered selective by the addition of dye brilliant green. Enrichment culture medium. Enrichment medium is liquid media, used to increase the relative concentration of certain microorganisms in the culture prior to plating on solid selective medium. It is mainly used as broth medium. Example: selenite F broth, tetrathionate broth, alkaline peptone. Water are used to recover pathogens from fecal specimens. Differential indicator medium. In this medium, dyes, metabolic substrates etc. are added so that the bacteria that utilize them appear as differently colored colonies. Such media are called differential media or indicator media. Differential media allow the growth of more than one microorganism but with morphologically distinguishable colonies. Examples: 1. Mannitol salts agar. Mannitol fermentation equals gram-positive. Bacteria Staphylococcus aureus produces yellow colonies with yellow zones. 2. Blood agar. Various kinds of hemolysis, i.e. A, B and G hemolysis. 3. McConkey agar. Lactose fermenters, pink colonies, whereas non-lactose fermenter produces pale or colorless colonies. 4. TCBS, Vibrio. Cholerae produces yellow colonies due to fermentation of sucrose. Transport media. These media are used when specimen cannot be cultured soon after collection. This media prevent drying, desiccation of specimen, maintain the pathogen to commensal ratio and inhibit overgrowth of unwanted bacteria. Example: Carey-Blair medium, Amy's medium, Stewart's medium etc. Stewart's and Amy's media are semi-solid media in consistency. Addition of charcoal serves to neutralize inhibitory factors. Carey-Blair transport medium is used to transport feces from suspected cholera patients. Pike's medium is used to transport streptococci from throat specimens. Anaerobic media. Anaerobic media is mainly supplemented with hemming and vitamin K where bacteria need low oxygen content, reduced oxidation reduction potential and extra nutrients for their growth. The media is also contains 1% glucose, 0.1% thioglycolate, 0.1% ascorbic acid, 0.05% cysteine or red-hot iron filings. Before use, the medium is to be boiled in water bath to expel any dissolved oxygen and then sealed with sterile liquid paraffin. Example: Robertson cooked meat, RCM. Medium is commonly used to grow Clostridium sppс. Contains a 2.5 cm column of bullock heart meat and 15 ml of nutrient broth. Thioglycolate broth contains sodium thioglycolate, glucose, cysteine. yeast extract and casein hydrolysate. Assay media. These media is used for assay of amino acids, vitamins and other antibiotics. Antibiotic assay media are used for determination of antibiotic potency by microbial assay. Raw materials and nutritional requirements for culture media. Raw materials play an important role in media preparation. Quality of media depends on the quality of raw materials. The most important raw materials are used for preparations of media are water, agar, peptone, casein hydrolysate, meat extract, yeast extract and malt extract. Water as solvent. Water plays a vital role in preparation of media. Water should be free from copper ion because copper ion inhibits the growth of the microorganisms. Petri dishes. Petri dishes are generally sterilized with ethylene oxide, ETO, or gamma irradiated. Only borosilicate glassware should be used because soda glass can leach alkali into the media. Energy source. The most common substance added to culture media is glucose. That acts as source of energy and also increases the rate of growth of organisms. Nutrients. The nutrients of culture media are selected to recover the required spectrum of organisms in the sample, e.g. coliforms or anaerobes. Proper nutrients help for bacterial growth. Essential metals and minerals. The inorganic essential components of culture media are divided on a semi-quantitative basis: macro components, GM, liter, Na, K, Cl, P, S, Ca, Mg, Fe. Micro components, MgM, micro GM, liter, zinc, Mn, bridge, B, Cu, company, Mo, V senior, etc. Buffering agents. The use of buffer compounds at specific pK values is especially necessary when fermentable carbohydrates are added as energy sources. Such examples of buffering agents are phosphates, acetates, citrates, zwitterion compounds and specific amino acids that added to culture media. Indicator substances. The addition of colored indicator substances in the medium is very effective of detecting fermentation of specific carbohydrates. They change color distinctly and rapidly at critical pH values. Examples: phenol red, bromocresol purple, fuchsin, etc., are toxic, and it is essential to use in low concentrations. Selective agents. Chemicals or antimicrobials are added to culture media to make them selective for certain microorganisms. They are added at specific concentrations to suppress the growth of unwanted organisms in a polymicrobial sample. Examples: bile salts, dyestuffs, tetrathionate, tellurite and azide. Antimicrobials are more specific in their selective action. Gelling agents. Gelatin is still used for a few specific media and carrageenans, alginates, silica gel and polyacrylamides are sometimes used as gelling agents, but the most important gel-forming substance used in culture media is agar. It is inert to microbial action and melting temperatures, 38 degrees Celsius and 84 degrees Celsius respectively. The high gel strength which allows low concentrations of agar to be used. It has low toxicity and also not hydrolyzed by agarase enzyme in high temperature. Microbiological. Agar is specially processed to yield a low toxicity, high clarity, low mineral and high diffusion gel. Other components. Many other substances are added to culture media for specific purposes like growth factors for fastidious organisms, pH-reducing compounds for anaerobic organisms. Thioglycolate and cysteine, whole blood to detect hemolytic enzymes etc. Meat extract. It is obtained by hot water extraction of lean beef and then concentrated by evaporation. It contains gelatin, albumoses, peptones, proteoses, amino acids, creatinine, purines, and accessory growth factors. Yeast extract. It is prepared from washed cells of bakers' yeast and contains wide range of amino acids, growth factors and inorganic salts. Malt extract. It is prepared by extracting soluble materials from sprouted barley in water at 55 degrees Celsius and concentrated by evaporation. It contains maltose, starch, dextrin, glucose and small amounts of protein and protein breakdown products and growth factors. Nutrition required for bacterial culture medium. Major macronutrients. Carbon, the main source, is organic compounds and CO2. It is the main component of cellular materials. Hydrogen, the main source is organic compounds. It is the main component of cell water. Oxygen, the main source is organic compounds, CO2 and O2. It is the main constituent of cell material and cell water. It is electron acceptor in aerobic respiration. Nitrogen, the main source is organic compounds, NH3, NO3, N2 etc. It is the main constituent of amino acids, nucleic acids, nucleotides and coenzymes. Sulfur, the main source is organic sulfur compounds. It is the main constituent of some amino acids like cysteine, methionine, glutathione and several coenzymes. Phosphorus, the main source is inorganic phosphates. It is the main component of nucleic acids, nucleotides, phospholipids etc. Potassium, the main source is potassium salt. It is the main component of cellular inorganic cation and cofactor for certain enzymes. Magnesium, the main source is magnesium salt. It is the main component of inorganic cellular cation, cofactor for certain enzymatic reactions. Iron, the main source is iron salt. It is the main component of cytochromes and certain non-heme iron proteins and a cofactor for some enzymatic reactions. Calcium, the main source is calcium salt. It is the main component of inorganic cellular cation, cofactor for certain enzymes and a component of endospores. Manganese, the main source is manganese salt. It is the main component of inorganic cellular cation, cofactor for certain enzymes. Major micronutrients. They are also known as trace elements because they require very small amount. They usually act as cofactors for essential enzymatic reactions in the cell. They are cations requires for bacterial nutrition. Examples: Mn, company, zinc, Cu, and Mo. Zinc 2 is present at the active site of some enzymes but is also involved in the association of regulatory and catalytic subunits in E. coli aspartate carbomoyl transferase. Mn2 aids many enzymes catalyzing the transfer of phosphate groups. Mo2 is required for nitrogen fixation and Co2 is a component of vitamin B12. Cu2 is an important micronutrient required as a redox cofactor of multiple proteins. Carbon and energy sources for bacterial growth. A source of carbon as energy source and other nutrients as growth parameters are required in a permissive range of physical conditions viz. O2 concentration, temperature, and pH. Sometimes bacteria are referred to as individuals or groups based on their patterns of growth under various chemical or physical conditions. For example, phototrophs are organisms that use light as an energy source. Anaerobes are organisms that grow without oxygen. Thermophiles are organisms that grow at high temperatures. Organisms that use light energy is called phototrophs. When organisms use an organic form of carbon are called heterotrophs, and when organisms that oxidize inorganic compounds are called lithotrophs. The source of carbon is either organic carbon or CO2. Organisms that use organic carbon are heterotrophs, and organisms that use CO2 as a sole source of carbon are known as autotrophs. Growth factors. All the bacteria require small amounts of organic compounds for growth because they are essential substances that the organism is unable to synthesize from available nutrients. Such compounds are called growth factors. They are organized into three categories. 1. Purines and pyrimidines. They are required for synthesis of nucleic acids, DNA and RNA. 2. Amino acids. They are required for the synthesis of proteins. 3. Vitamins. They are needed as coenzymes and functional groups of certain enzymes. Some bacteria, e.g. E. coli, do not require any growth factors. They can synthesize all essential purines, pyrimidines, amino acids and vitamins, starting with their carbon source, as part of their own intermediary metabolism. Certain other bacteria, e.g. lactobacillus, require purines, pyrimidines, vitamins and several amino acids for their growth. Mutant strains of bacteria that require some growth factor not needed by the wild type parent strain are referred to as auxotrophs. Physical factors affecting bacterial growth. Nutrient concentration. Bacterial growth is directly proportional with the available nutrients. If culture media is rich in nutrients, growth of bacteria occurs faster and vice versa. With increase in concentration of nutrients, growth rate of bacteria increases up to certain level and then growth rate remains constant irrespective of nutrition addition. Temperature. The lowest temperature that allows the growth is called minimum temperature, and the highest temperature that allows growth is called maximum temperature. There is no growth below minimum and above maximum temperature. Below minimum temperature, cell membrane solidifies and becomes stiff to transport nutrients into the cell, hence no growth occurs. Cellular proteins and enzymes denatures at maximum temperature, so the bacterial growth ceases. Growth rate of bacteria increases because the rate of metabolic reaction increases with increase in temperature. At certain temperature the growth rate become maximum. This temperature is known as optimal temperature. On further increasing the temperature above optimal, growth rate decreases drastically and completely ceases with reaching maximum temperature. Thermophile organisms are heat-loving bacteria. Their optimum growth temperature is between 45 degrees Celsius and 70 degrees Celsius and are commonly found in hot springs and in compost heaps. Hyperthermophile bacteria grow at very high temperatures. Their optimum growth temperature is between 70 degrees Celsius and 110 degrees Celsius. They are usually members of the archaea. Psychrophiles are grown between -5 degrees Celsius to 20 degrees Celsius. Mesophiles are grown between 20 degrees Celsius to 45 degrees Celsius. pH. pH affects the ionic properties of bacterial cells, so it affects the growth of bacteria. Most of the bacteria grow at neutral pH, 6.5 to 7.5. However, there are certain bacteria that grow best at acidic or basic pH. Microorganisms can be placed in one of the following groups based on their optimum pH requirements. 1. Neutrophiles grow best at a pH range of 5 to 8. 2. Acidophiles grow best at a pH below 5.5. 3. Alkaliphiles grow best at a pH above 8.5. Ions and salt. All bacteria requires metal ions such as K, Ca, Mg, Fe, zinc, Cu, Mn, etc. to synthesize enzymes and proteins. Most bacteria do not require sodium chloride in media and they can tolerate very low concentration of salt. Some halophilic bacteria such as archaeobacteria require high concentration of salt in media. It is balanced with osmosis process. Gaseous requirement. Oxygen and carbon dioxide are important gases that affect the growth of bacteria. Oxygen is required for aerobic respiration and obligate aerobic bacteria must require O2 for growth. For example, Mycobacterium, Bacillus. For obligate anaerobes, oxygen is harmful or sometimes lethal, but facultative anaerobes can tolerate low concentration of O2. Carbon dioxide is needed for capnophilic bacteria, such as Campylobacter, Helicobacter pylori. Water is the most essential factor for bacterial growth. Available water in the culture media determines the rate of metabolic and physiological activities of bacteria. Sugar, salts and other substances are dissolved in water and are made available for bacteria. Preparation of nutrient agar. 1. 28 grams of nutrient agar powder is suspended in 1 liter of distilled water. 2. Heated the mixture while stirring to fully dissolve all components. 3. Autoclaved the dissolved mixture at 121 degrees Celsius for 15 minutes. 4. Allowed it to cool but not solidify. 5. Poured nutrient agar into each plate and left the plates on the sterile surface until the agar has solidified. 6. Stored the plates in a refrigerator. Uses of nutrients agar. 1. It is frequently used for isolation and purification of cultures. 2. It is also used for producing the bacterial lawns needed for antibiotic sensitivity tests. I request to you, please subscribe this channel and like and share this video to get more videos and updates. Thank you.